Let's have a quick summary of fibronectin, which we have already learned during our previous video. So, fibronectin is a protein, is a linear array of distinct building blocks that give each polypeptide a modular construction. Each fibronectin polypeptide is constructed from a sequence of approximately 30 independently folding fibronectin modules, two of which are depicted. And while FN type modules were first discovered in fibronectin, they are found as part of many other proteins also. From blood clotting factors to membrane receptors, presence of shared segments among diverse proteins strongly suggests that many present day genes have arisen during evolution by a fusion of parts of separate ancestral genes. So there is an ancestral linkage involved. In fibronectin, the 30 or so structural modules combine to form 5 or 6 larger functional domains. There will be larger functional domains and they are connected. Each of the two polypeptide chains that make up a fibronectin molecule contains binding sites for numerous components of the ECM. The two fibronectin molecules, the polypeptide chains of there are two polypeptide chains that make up a fibronectin molecule. So they contain binding sites for numerous components of the extracellular matrix such as collagens, proteoglycans and other fibronectin molecules. These binding sites facilitate interactions that link these diverse molecules into a stable interconnected network. Binding activities of fibronectin molecule are enhanced by mechanical forces that pull on these fibrous proteins and unfold its component modules. Unfolding of these modules expose binding sites that work otherwise have remained buried. So there are some binding sites which are acting as a binding site for numerous components of the ECM such as collagen, proteoglycans and other fibronectin molecules. These binding sites facilitate interactions that link these diverse molecules into a stable interconnected network. Binding activities of fibronectin molecule are enhanced by mechanical forces that pull on these fibrous proteins and unfold its component modules. Unfolding of these modules expose binding sites that would otherwise have remained buried. So some other points are also there, like the binding sites for receptors on the cell surface. First binding site was for numerous components of ECM, collagen, proteoglycan and other fibronectin molecules. The second binding site for receptors on the cell surface. These binding sites hold the ECM in a stable attachment to the cell. The importance of fibronectin containing substratum for cell attachment can be seen by some micrographs. What is the importance of fibronectin and other extracellular proteins? That can be easily recognized from the example of embryonic development. Development is characterized by waves of cell migration during which different cells follow different routes from one part of the embryo to another. So migrating cells are guided by proteins such as fibronectin. So that is the importance of fibronectin.